Let's talk about natural selection and microevolution. In the last video, we defined what evolution was, as well as microevolution and macroevolution. Microevolution being changes in a gene pool in terms of allelic frequencies, and that might represent or be seen as phenotypic changes in the population, but doesn't result in new species overall. Whereas macroevolution is when we have changes in the gene pool at such a large scale that it creates new species. In terms of natural selection and microevolution, we did discuss briefly that natural selection is the process that leads to these microevolutionary changes. And in this video, we're going to be talking about how variation, natural mutations of variation in a population can then lead to changes in the gene pool. In the next video, we'll be looking at how natural selection selects and the three different types of phenotypic selection. So variation, you probably recall, we've talked about this plenty of times before, but variation exists within a population just naturally. And a great example of this is humans. If you look at human populations, there is a huge amount of variation. So just looking at your family, looking at the people either side of you in class, you will see naturally variation in height, variation in other size aspects, variation in terms of hair color, eye color, skin tone, all of that. Now, in any organism, you're going to see different phenotypes and variations within that population due to differences in the genotypes. And those variations can be inherited by the offspring and passed down to the next generation. In the wild, when natural selection is acting, some of those variations, or in some cases they result from mutations, might give an advantage to survival. And if you can survive better, you can reproduce better. And that variation will then be effectively selected for moving forward. Peppered moths is an example you've heard, I'm sure, a dozen times before, and it is a great example of this. We'll talk about that in greater detail in a moment. But when we're talking about the gene pool, and we mentioned this briefly in the last video, but gene pool is the range of genes and all the alleles present in a population. So if I was to have a population of 100 gorillas and I was to know their alleles for a particular trait of all of those 100 gorillas, then that would be my gene pool. And when we look at the population, the variation in the population is restricted by the alleles available in the gene pool. If we've got in the gene pool genes for black hair and we've got genes for brown hair, we're not all of a sudden going to have an individual with pink hair unless a mutation occurs. So in the absence of a mutation, variation is restricted to what already exists within the gene pool. But if a mutation occurs, it can add new alleles or genes to the gene pool of a population, and this would be an interesting thing in terms of selection. Peppered moths, we know they come in the dark variety, we know they come in the light variety, and every individual that's born in that population, in the absence of a mutation, will be one of those varieties. A yellow peppered moth, for example, would only occur through a chance mutation. So when we look at how this works in terms of adapting to the environment, the peppered moths is a great example. As I said, we'll go through it. So prior to the Industrial Revolution, when there was no pollution and whatnot, then the majority of the peppered moths were light, as you can see in that graph. Okay, they captured moths across that whatever study that was, and they found that the majority of the moths were light in color. The reason being is that there was lichen all over the tree trunks, and the lichen was that sort of light white, sort of speckled gray color, and so the light individuals could camouflage well. Okay, so they were effectively being selected for. At the same time, there were dark moths already existing in that population. So within the gene pool, we didn't just have light moth alleles. We also had dark moths alleles. But in the pre-industrial revolution environment where being light colored was an advantage, then the allele for light coloring was much more frequent. It had a higher allelic frequency. After the Industrial Revolution, we can see in the change in the graph there, the dark coloring, the dark phenotype was advantageous. And so what happened was more light moths were getting eaten before they could pass on their trait. And you can see that demonstrated here as well. So the blackening of the tree branches meant that predators could find the light-colored moths more easily, and the dark-colored moths could survive better because now they could camouflage better. So what that meant was the light-colored moths were more likely to die before they could breed and pass their genes on to the next generation. The dark moths were more likely to contribute to the next generation and pass on their genes for dark wing and body color. So the result of this is only the dark moths were reproducing in huge numbers because the light moths were getting killed. And so there was an increase in the frequency of the dark allele within the population. Because there was an increase in the frequency of the dark allele, then that resulted in the next generation, a greater number of dark moths, and so on. And as this kept happening, generation to generation, we saw a shift where there was a decrease in the frequency of the light allele and an increase in the frequency of the dark allele. So the allele for dark pigmentation in these moths, let's say, is capital A, and that's dominant over the light coloring, which is lowercase a. So the moth population contained some individuals with a dark phenotype carrying the big A allele. During the peak of the Industrial Revolution, the big A dominant allele became more common in subsequent generations, increasing its allelic frequency. And the recessive trait for light coloring became less common. So this is a perfect example of microevolution because we have not got a new species of peppered moth. All that's happened is the population has changed in regards to one trait as a result of a shift in allelic frequency within a gene pool. So as we know now, and as I've just said, microevolution is a change in the allelic frequency or the gene pool of a population. And that is going to happen over, the course of a couple of generations to many generations. For any of this variation to occur, for microevolution to show a change in allelic frequency, then we have to have more than one allele to begin with, which is the case in most things. Phenotypes that vary because of genetic differences are called genetic polymorphisms. So a light-colored moth and a dark-colored moth, that's a genetic polymorphism. And as I said, this is a lot of traits. The allelic frequency is not always equal. In fact, it's almost never equal. And the fact that that is the case is what changes with microevolution. So frequency of alleles in a population change in response to these four main things. So mutation of a new allele introduces a new allele to the population or the gene pool. So that changes the allele frequency because now instead of looking at, how many of just X and Y, you've now got Z thrown into the mix. Immigration into the population. So new migrants moving into a population can affect the frequency of alleles, as well as emigration out of the population can affect the frequency of alleles. You see in this image here, we've got a population of green beetles and a population of brown beetles. That if a brown beetle enters the green beetle population, then all of a sudden the frequency of the brown allele has increased. Okay, and vice versa. If you only had one or two brown individuals in a predominantly green population, and then those brown beetles decide to leave, then that's going to significantly drop the allele frequency of brown alleles in that population. As well as that, there's different rates of reproduction of different individuals or their fecundity. And that can be as a result of them dying young and not being able to reproduce, or it can be due to random factors, which in some populations might mean that some individuals just happen to naturally not have high fecundity. Our basis of natural selection is that favorable traits will become more common in successive generations as they're selected for. So those that contain the less favorable alleles are less likely to survive and reproduce, and therefore their alleles will be reduced in frequency. Or conversely, those that have the more favorable alleles, like the black moths during the industrial evolution, will be able to survive better, survive longer, reproduce more effectively, and in greater numbers. And so the over time, the overall allelic frequency of the gene pool changes. The population is now blacker than they were before, as opposed to being predominantly white, and they're better adapted to their habitat. Mutations often produce recessive alleles, which can be masked by the original allele. So they're adding to the gene pool in that they're introducing a new allele, but they tend to remain at low allelic frequency unless the environment changes and they may become more useful and therefore selected for. There is a high likelihood of mutations occurring and then just silently disappearing in future generations if it never gets selected for and other factors select against the individual with that mutation. So they can just be silent and disappear over time. And that likely happens all the time without any notice because the mutations are of low consequence. So in terms of migration, immigration, and emigration, we talked about before. So when we're talking about gene flow, we're talking about alleles transferring between different populations. So moving from one population to another, and that results in changes in the gene pool. Okay, so as it says there, gene flow is the transfer of alleles that results from emigration or immigration of individuals between populations. So migration between different populations is going to occur, and we can end up with an exchange of alleles there. It's highly unlikely that the individuals coming in and the individuals going out are going to equal out in terms of their alleles. So immigrants moving in can introduce brand new alleles to the gene pool, or at the very least, they're going to increase the frequency of whatever alleles they contain. Immigrants that leave the population could completely remove some alleles, or at the very least, they're going to reduce the frequency of the alleles they contain. So either of these can cause significant changes to the gene pool over time, depending on how these processes happen. We have a couple of other factors that can change allelic frequency as well. We're not just talking about immigration and emigration. There's these three here: genetic drift, bottleneck effect, and founder effect. So genetic drift refers to a change in a population that is completely random. It typically only occurs in small populations because of the sort of randomness of this, chance change in the gene pool is not noticed in a large population. If we had some genetic drift occurring in a population of krill where there's millions of them, it's not going to be noticed. Whereas if you've got an almost extinct population where genetic drift occurs, then it's going to have a significant effect. So each of us is getting half alleles from each parent. Any organism that sexually reproduces works thusly. So which half of their alleles we inherit is completely random, and also the sex and amount of offspring can be random. So even though you might have a parent that has, let's say in the example in the diagram here, two lowercase b alleles, okay, depending on what you inherit from your parents, it might just so happen that over the case of one generation, the population of a particular allele drops significantly through random chance and mating, okay? Or it could end up that those traits end up with females that are not fertile or something like that. There's lots of randomness that can occur that can contribute to this. And as I said, it's not felt in a large population. It's an insignificant change. But in a small population, the randomness could mean that some alleles are completely lost. They're removed from the gene pool. If it increases the number of an allele, then that's not going to be as noticeable. But it could result in a trait being completely removed from the gene pool altogether. So that random change can have a significant effect. Bottleneck effect we will talk about in greater detail when we get towards the end of the evolution unit. Bottleneck effect occurs when some sort of catastrophic event or period of bad environmental conditions, earthquakes, we're talking about tsunamis or any catastrophic event that occurs, reduces the population drastically. And what then happens is with that population crash, we potentially lose a lot of the alleles that were adding variation to the gene pool. So certain alleles can be completely lost through chance, reducing the gene pool as they can no longer be recovered. Okay, if we completely get rid of all individuals with a particular trait, and they're the only ones who have the alleles for that trait, we can't get it back. I mean, there's a very minor chance it could randomly mutate, but it's unlikely to occur. Okay, and the surviving alleles are strictly the advantageous ones in this case. They were the ones that allowed them to survive through that natural disaster. But because we've now got a small population, it's susceptible to genetic drift. This bottleneck effect has been seen in cheetahs. They have incredibly low genetic diversity because of a bottleneck that happened a long time ago. So that means that they're susceptible to genetic drift. They're susceptible to not so favorable changes in gene pools. And Tasmanian devils are the same. The founder effect is the sort of last thing that we're going to look at in terms of changing these gene pools. And the founder effect occurs when a small group of individuals break off from a larger population to establish a colony. And what happens is that new colony can only get its alleles from the existing gene pool. So the gene pool of that new population is less diverse, and it's made up of only the alleles the founders have. It's similar to the bottleneck effect, except it has a different mechanism. And depending on who you ask, there's an example of, I think it's called Pipalap or something like that. It's an island in the Pacific that is called the Island of the Colorblind. One of the reasons is that, and again, depending on who you ask, it's either a bottleneck effect or a founder effect. There was a massive cyclone that came through and wiped out most of the population. There was only, I think, about 40 people left in the entire island, and one of them was the mayor of the island. Now, the mayor was obviously a powerful man, very desirable. He just also happened to be a very rare form of basically complete colorblindness, where everything was gray to him. Now, he, being the man he was, took it upon himself to repopulate the island after this massive event. And so, let's say he had kids with 3 or 4 of the local women, then he was passing on his colorblindness genes to the children of these 3 or 4 women, where in an ordinary circumstance, he probably would only be having kids with 1 woman. And so as a result, that increased the allelic frequency of this colorblindness gene. Now, it didn't actually affect them being colorblind in this circumstance. On the island where they lived, they had lots of creature comforts and color didn't matter too much. And so it didn't get selected against. And because of the increased frequency of it in the population, that meant that over time, the island has become progressively more and more colorblind due to, again, depending on who you ask, either the bottleneck effect or the founder effect. Some great examples there of those 2 effects, but we will leave it there. When we come back with the next video, we'll be talking about natural selection and patterns of selection.